people have talked on and off through the week about the RNP delivery as a beneficial alternative to message delivery or DNA vector delivery. And so I wanted to just take it one small step farther and talk about some of the data in the literature and that we've accumulated at IGI that support this idea. Okay? So the very first paper that used RNP delivery for genome editing with the CRISPR platform was this one. And the organism was not a 293 cell. The organism was the nematode that Barbara had talked about. And the main reason for doing this particular experiment was that in the nematode, as some of you may know, the worm suppresses expression of transgenes, particularly in the germline. And the whole phenomenon of RNA interference was discovered by Andy Fire and Craig Mello by injecting messenger RNAs into the worm germline and finding that not only wasn't the message, the synthetic message expressed, but that synthetic message suppressed the expression of the endogenous gene that corresponded to it. And the reason for that was there was a little bit of antisense RNA in the mix with the messenger RNA, and it kicked off RNA interference. And so in my lab, we actually applied the zinc finger nucleases to C. elegans very early on, and were never able to get germline mutagenesis. We could get somatic mutagenesis in spades, but we couldn't get it in the germline. And Barbara's group was the first to really get the ZFNs and the talons and then the CRISPRs to work in the C. elegans germline. But I thought that it was a neat idea to try injecting the Cas9 RNP, because then you're not dependent on translation at all. And you don't care, even if your little RNA is, kicks off some RNA interference, there's nothing to interfere with. And you're just putting the active nuclease in directly. So I'm a co-author on this paper. The experiments were actually done in Korea by some collaborators. But this is just showing that you can make a synthetic guide RNA, purified Cas9 protein, put them together, inject them into the worm gonad, just the way Barbara was illustrating, and then for two different targets, these are just individual injected worms, but just from 9 injected worms, 1, 2, 3, 4 of them gave mutant offspring, which you could score just in the next generation. And after Barbara's talk, I went back and I looked to see what the targets were. And you remember she was emphasizing this 3' GG next to the PAM. Well, one of the guides had that, and the other one didn't. And this one was maybe a little more effective than the other, but and as she showed, not all non-GG guides failed. So this is at least consistent with that. Anyway, and this is just showing the dumpy phenotype and the uncoordinated phenotype for these two mutants. So what people have done in mammalian cells is something very similar to make purified Cas9 protein, synthetic guide RNA of some kind, put them together with or without a repair template for homologous repair, and a lot of people are doing nucleofection or some other kind of electroporation into cells. This picture is from the paper from Jennifer Doudna's lab, that top reference there. And Jinsoo Kim, in addition to helping out with that worm paper, also applied the Cas9 RNP to human cells, and this is just showing different concentrations of the guide RNA and of the Cas9 protein. And for reference, I've shown how many micrograms correspond to how many picomoles, because as it turns out, in making the RNP, it's pretty important to have at least something of an excess of RNA over protein. If you don't do that, you tend to get some aggregation of the protein. And so I think these are all situations where there was a good excess. I'm sure there was good excess of RNA over protein, and showing that they could get non-homologous end joining mutagenesis efficiencies comparable to plasmid delivery, and showing that they could do it in a number of different genes. And I think this was all in K562 cells. And then from the same paper, they're looking at some human primary and ES lines, and showing that, amongst other things, that these sensitive cells survive better, having been targeted at 2 different locations within the genome. They survive better and retain their colony forming ability when they're RNP transfected, as opposed to being DNA transfected. So, in addition, this is an off-target analysis from that same paper. They're a little hard to credit at first glance, because they're using a log scale for the indel frequencies. But in each case, they're showing that the ratio of on-target to off-target mutagenesis is better for the RNP delivery, which is in blue, as compared to the plasmid delivery, which is in green. And the on-target efficiency suffers very little by using the Cas9 RNP. In fact, many people find the Cas9 RNP is better for them at the on-target mutagenesis. One of the advantages to the RNP delivery is that the hit and run is much quicker. That you're introducing the nuclease itself rather than waiting for transcription of a plasmid and/or translation of a messenger RNA. So, looking at indel formation as a function of time after initiation of delivery, the RNP is giving indels that you can detect very early on, and it's plateaued by 24 hours. And monitoring the protein by Western blotting in these cells, you see that the protein itself is pretty much gone by 24 hours. So a couple of advantages here. One is that you're getting things happening very quickly, and the other is that reagent isn't around for a long time to continue to hit secondary targets, even after maybe you've done as best you can at the design target. So these are some advantages to doing the RNP delivery. This is a figure from the Doudna lab paper on this, where they're targeting this particular exon of the EMX1 gene with a long oligonucleotide that has an insertion in it that carries a HindE3 site that they're going to assay down here. And there's a lot going on in this figure. First thing to say is that they're getting good overall percent total editing. I think that's what the percent TE is for, percent total editing. So this is a T7E1 assay, and both the homologous and the non-homologous mutagenesis would show up in the T7E1 assay. So with different amounts of the Cas9 protein, of the Cas9 RNP, and in these various treatments, they're getting high levels of total editing, and also getting fairly reasonable levels of homologous repair, as judged by the sensitivity of the PCR product across this to HindE3 digestion. And what's going on along the top is treatment of the cells with various cell cycle inhibitors. Fyodor Ernov this morning said that at Sangamo, they right from the start decided to use a cell cycle inhibitor to try to arrest cells in a cell cycle phase that would be favorable to homologous repair. And they used vinblastine, which also arrests cells here at around the G2M border. And in the Doudna lab, they found that using nocatazole, which is arresting here, that they could get an improvement of the HDR efficiency over some of the other treatments. And it's not a huge improvement, but there's something. There are other cell cycle inhibitors you can look to see. There are several that were inhibitors within or within S phase or at the G1S boundary, and those were the ones you might have expected would be the best. So the way the experiment's done is that you arrest the cells for some period of time. Usually it's one cell cycle. With one of these treatments, you have to sort of double arrest them in order to get them really synchronized. Then you deliver the Cas9 RNP and then release them from the arrest for a subsequent culture. So you might have thought that the best treatments would be the ones that line the cells up at the beginning of S phase, because S phase and G2 are where homologous repair is supposedly the very best. But as it turned out, it was this nicotazole treatment that had an effect on these 293 T cells. So there's a little more information just looking at concentrations of the template for repair, that single-stranded oligonucleotide template. They look at plus and minus strand versions of that template, and they're still looking at the RNP concentration that goes in. And I think over here they're up in the 30% range, 30% and above range for the percent of alleles that are homologously repaired. So that's a pretty high efficiency, and that's all using nicotazole treatment to boost things above the level of untreated cells. And those were, HEK 293 cells, long-term adapted to culture. When they went to primary cells and to these human embryonic stem cells, the frequency of homologous repair in these cells is really pretty lousy if you don't do anything to them, even if you're doing RNP delivery. And for some of the situations, the T7E1 assay is giving you fairly reasonable levels of indels, but you never get anything very good for homologous recombination. And in this situation, nicotazole alone has no detectable effect, and they did a sort of a fancy treatment with nicotazole for a while and then acetylcholine for a while, sort of hoping to get some sort of combined benefit. But the frequencies of homologous repair down here are in the 1% to 2% range. So there are some advantages, some very measurable advantages to the RNP delivery. You're getting high levels of total editing and a pretty good percent of homologous repair. But in many cell types, that the benefit for homologous recombination really isn't all that helpful. So other things are going to have to be done. I'm going to now show some data from a paper that came out of Thermo, the Thermo Fisher sort of research and development lab. And the data I'm going to show are some of the ones that encourage those of us at IGI to begin using RNP delivery because as you'll see in a table in just a minute, the efficiencies they were getting across a number of different cell lines were really impressive. And the paper's finally been published. But one of the things they did, and the Doudna lab did something very similar to this to produce the guide RNAs, was to make a long constant, actually I think they cloned this or made it as a PCR product, a long constant region for the constant region of the single guide RNA, and then used two shorter RNAs two shorter oligonucleotides, DNA oligonucleotides corresponding to the new guide sequence, and then a primer up here that corresponds to the T7 promoter, and a small primer down here to just amplify everything all together. So you put this together with that, and that, and put it into a thermal cycler, and it makes a nice double-strand template for in vitro transcription by T7 RNA polymerase. And one of the things we learned from these people was that somebody earlier was talking about the fact that they always anticipated they would get 2 Gs. They needed to have 2 Gs at the 5' end of their guide RNAs if a T7 promoter was being used. One of the things we learned from the people at Thermo is that you only need to have 1 G there, although a lot of the literature says that the T7 promoter goes all the way out to another G beyond that one and needs those, and those Gs appear in the transcript at the 5' end of the transcript. Making designs like this where there's only 1 G only 1 G in this position, that G will appear at the beginning of the transcript, but there's only one of them. Okay. All right, so they can make the templates, they can make the RNAs, and then they apply RNP delivery to a lot of different cell types. The RNP delivery is over here either by liposuction or by electroporation using the thermo-produced electroporation machine, and then comparing this to plasmid delivery or mRNA delivery of the Cas9 protein. And this table was on a poster at a meeting that we went to in January this year. And what we were looking at is this right-hand column and going, that's amazing. We got to do this. And they're using human cells, mouse cells, stem cells. They're using cord blood cells. They're using a variety of different cell types and always getting really good levels of end-join mutagenesis. So there's no donor in these experiments. This is just end-join mutagenesis. And then they show that they can hit 2 or 3 targets simultaneously just by assembling the RNP with 2 or 3 different RNAs, just a mix and assemble process. And you can see that if they use the T7E1 assay on the cells from the mixed electroporation, that each of the target genes is very effectively mutagenized, and looking at one alone, the other alone, or dual targeting, they're getting double knockouts in both genes. So biallelic knockout for both of the target genes at very high frequency. And this one's a little more complicated because they're hitting 3 genes, but they're getting high levels of triple biallelic knockout in that cell line. Actually, they're getting. I think these are male cells, and one of the targets is HPRT, which is on the X chromosome. So it's only 5 alleles that they're hitting. Yeah, they shouldn't brag, right? But the point of this is just that the RNP delivery is being very effective. So I'm now going to show you just a couple of slides from IGI and the project that I've been working on. So the way we do this is we order one long oligonucleotide that's constant from IDT, and 2 short ones outside, and then we put the guide sequence into an oligo that, depending on the length of the guide sequence itself, is on the order of 57, 58, 59 nucleotides. So why is that important? Well, for IDT anyway, the price jumps about 5-fold if you go above 60 bases in the oligo. So we're now ordering standard oligos with our new guide instead of these ultramers that cost quite a bit more. So again, the T7 promoter is out here, and the very short oligos you use in large quantity in the amplification to make the PCR template, and each of these oligos you use in much smaller quantities. And one of the things I did was I shortened the overlap here. People were often using a 20 nucleotide overlap. By making it 15 instead, that helped to bring things down under 60 nucleotides. And You know, if you're doing PCR with purified oligos, the TMs almost don't matter. The TM of this overlap is something like 15 or 20 degrees lower than the TMs of the matches for the outside primers, and it just doesn't matter because you don't need to get very many extensions from this middle overlap to have a template for the outside ones, and once they get going, you know, you're off and running. So. Just doing a 1 hour PCR, you don't, you know, it's such a short product, you don't, you know, the time is very short. You can get a couple of micrograms of this T7 template out of a 20 microliter reaction. And then we just transcribed these in vitro with T7 RNA polymerase. This is running the DNA template on a gel, nice and clean, and this is running the RNAs on a polyacrylamide urea gel. And the RNAs that come out are really clean, really nice. And you can get up to a couple of hundred micrograms off 120 microliter reaction. And the way we've been doing it is well, I can do it here, we just take some of this DNA product out of the tube it was made in and put it into a different tube with the T7 transcription reaction components and put it back in an incubator at 37 degrees for, actually you can let it go for kind of a long time. I don't know what we're doing in the lab right now, but we were doing overnight incubations for the T7 transcription because, you know, the template's still there. As long as there are nucleotides there and the polymerase is active, it's just going to keep making more RNA for you. So we've been able to get high yields of RNA. And then this is just showing one of the applications we've made of the RNP delivery. So Mark DeWitt, the postdoc I've been working with in the lab, he and I designed and he produced a reporter construct that is an integrated BFP gene. There's a CMV promoter driving it. Sometimes we use a version that has a PEST sequence at the end, so the BFP protein is turning over rapidly, and our readout can be made in a short time period. And BFP and GFP differ by a single codon within their protein sequence. So we provide a template for repair that contains the GFP codon, and so we have a guide RNA that sits right on top of that codon, as close as we could. I mean, I think the break is almost in the codon, but it's very, very close. I didn't bring that slide, and so we can score loss of BFP because the blue fluorescence is going to go away and go into a different portion of the FACS analysis. And we can score homologous repair by incorporation of this codon that will convert this to GFP. So simultaneously, we're looking at end-join mutagenesis and homologous repair. So this is just telling things about how we made the reporter. This is FACS analysis before the RNP nucleofection, and 94% of the cells are BFP positive. You know, as you grow cells with a reporter like this, there's some silencing, some sort of loss of signal, but we've got cells that are quite stably producing BFP, no GFP, whatever, and a little bit of loss of the BFP signal just by having the cells in culture. And then with the RNP nucleofection for that target within BFP, we're getting, add these two numbers together, we're now at 99% BFP minus and 25% GFP plus. So the efficacy of this is in the HEK 293 T cells, the efficacy of this is enormous. And we were talking earlier, it's a little hard to give you a comparison to some of the DNA delivery we were doing before, because we hadn't really optimized that, and we sort of gave it up before we got it to be as good as it could be. But we were never getting anything even close to this. We were never getting as much as even 50% BFP knockout. So for us, the RNP delivery made a big difference.
I had a question about the days. Why did you take it to 6 days? Did you check earlier?
Yeah. One of the things we take it out for a while. This was in a cell that did not have the pest sequence on the BFP to destabilize it, and so you let the culture go for a while to make sure that the BFP protein is gone. I guess the messenger RNA has a half-life as well. So the conversion to GFP depends not only on the GFP protein being made, but also the loss of BFP. Okay, so and I guess the fact that there are no double positive cells essentially means that the BFP protein that was present is now really gone. And so this is just toting up the numbers. And then I showed you this slide before when I was talking about the T71 assay. And the reason I'm showing it to you now is because in another target that we were working with, now a genuine genomic target, we were testing different guide RNAs that were tiled or distributed over a small region of the gene that we were interested in. And the 2 4s and the 2 5s are effectively the same guide RNA, just slightly different versions. But in testing, I think there are 7 different guide sequences here being assessed by the T71 assay. This one really didn't work. And some of the others are fairly weak, but out of 7 guides tested, 5 of them clearly worked in this very small region using RNP transfection. So we're getting high frequencies of mutagenesis and with a lot of different guide RNAs using this procedure. And so this is the advertisement, right, for the RNP delivery. There are companies that are selling the Cas9 protein now. In Berkeley, we and shortly they are fortunate to have a core laboratory that will produce protein for us here. And so we've had the protein that we've used after a couple of trials with some loan from the Doudna lab. We've been using this core facility to produce the protein, and it's been very effective. But even in Utah, starting more than 2 years ago, there was a graduate student in a neighboring lab to mine who got a plasmid and a procedure from Jinsu Kim to make Cas9 protein out of E. coli, and succeeded with the first try. I think this protein had a his tag on it, so it was it was an induction, break open the cells, nickel column purification, and then use the protein, and it was highly expressed and easily purified. And one of the nice things about Cas9, which is not so true about the ZFNs or the talons, is that it's a bacterial protein, and E. coli will produce it with no argument. So, and then, with now these chemical syntheses of the guide RNAs. You can either save yourself some work by buying the guide RNAs, or make guide RNAs that have other features. The synthetic ones don't have to have any G on the 5' end because there's no constraint on their synthesis. Laura K. Brun was talking about doing chemical protection. As she showed, that doesn't seem to be as important for the RNP delivery, just because you're already protecting the RNA in the assembled RNP itself. But depending what your system is, and the use that you're trying to make. If you're just looking to make sort of short-term delivery and indels or HDR products, the RNP deliveries can be a real advantage. If you're looking to do broad screens, it's a little more challenging to do it that way because you know, it'll eat up more of the Cas9 protein. But people here and at UCSF that we're working with are looking into ways to possibly do an RNP-based multi-well type of screen. So just we've found this to be beneficial. As Jacob said, the lab's pretty much completely converted over, and happy to help anybody that wants to adopt it and has any difficulty with it. There was some interest in talking about donor design. So, so what were you particularly interested in? Yeah, so you're trying to do a translational fusion or something, right? So what you're looking, so this is the break, and what you're looking to do is to insert something that's pretty large. If you're going to do that, you can't really put it into an oligonucleotide. If you're going to put in, you know, a LOX-P site or something like that, that can go in an oligonucleotide donor with no real difficulty. You don't need more than a few tens of bases on either side of the break in order to make a good donor. Putting insertions in may drop the frequency of use of the donor a little bit, but it probably, the donor incorporation probably depends more on the efficiency of cleavage than it does on, you know, a little bit of insertion or mismatch in the donor. We've found in our hands that with any of these break-induced recombinations that mismatches in the homology arms between the donor and the target make very little difference. I mean, if you're making huge mismatches, that's a bad thing. But you know, the occasional mismatch here or there to inactivate a PAM or to, you know, some other way prevent cutting, those mismatches are tolerated fine. Doesn't decrease the level of homologous repair. So for something like this, you want to make a double-stranded DNA template that has homology arms of a few hundred base pairs at minimum, and you want, as Fyodor was emphasizing this morning, and I had talked about these conversion tracks before also, you want that break to be as close to the insertion site as possible. And the other thing is, if you want to make a deletion at the same time, maybe you want to put GFP in here, but you want to delete a bit of coding sequence upstream of that for some nutty reason, right? Okay. What you want to make sure is that one end at the break still has homology to the target. And I emphasize this, other people have talked about it. So you can copy this in, this is actually still continuous. You can copy this in as, from one side or the other. But if you can't copy it in from either side because you don't have homology close to the break, that's not going to work. So I'd say one other thing about donor design. Everybody will recognize this as a CRISPR target, a Cas9 target. So both strands of the DNA target are cut by Cas9. But one is RNA bound when it's cut and the other is not. This duplex is held pretty tightly in the Cas9 protein. It's actually binding that duplex. It's how it does recognition. It asks, is there a duplex there? And it's a little more ragged in holding onto it out here, and that's why mismatches are more tolerated out there. But there's an asymmetry in the strands here. And so even in that Doudna RNP paper I was talking about, people have used single strands of that polarity and single strands of that polarity for the same target. And the general observation is, doesn't matter. But there are people who are continuing to work on donor design. I saw a poster at that same meeting that I went to in January, where some people from Caribou, it's just down the road here, working sort of the holding company for Berkeley CRISPR technology. Somebody from Caribou was saying they had these odd sort of overlapping oligo donors that seemed to work as their best donors for a particular cut. And at that time, they'd really only used them at a couple of targets. I don't know how general that observation will be. But people are working on donor design either rationally, seeing if they can take advantage of this asymmetry, which, you know, to a first approximation, it's hard to do, or just looking to see, you know, if I make a lot of different donor designs, does something consistently work better than another? And one of the things that's a little frustrating in this area is that when people find something that works, they stop and they use it. So often the search for optimum tools is driven up to some point and then discontinued. And so often things that are found to work well in one system don't turn out to work particularly better in another system because whatever was found in one system isn't generalizable. Maybe it has something to do with the target rather than the donor. They're just things that people haven't studied in a systematic enough way to really come to a general conclusion. And it's sort of the same as with the rules for guide choice. You know, there are now algorithms that can help you with that choice, but there isn't any one simple rule that you can say, this is the way I'm going to design my guide RNA. And even with what Jacqueline was showing before, some of the, if my messages to Fifi come up, just ignore them. Even some of the poorly scoring guides were giving, you know, a very few of them were giving pretty high levels of indel formation. So we don't have complete control of this yet. It's great. Maybe we don't need complete control. Yeah, Chong?
A little bit more about the donor piece. Someone earlier in the week mentioned that on one side they saw that it went in and had homology as intended, and then on the other side it was unexpected where it seems like it was a blunt ligation too.
Right.
What was that about?
Yeah, so this is Randy's talk about zebrafish. And so years and years ago when Maria Jason was doing things with ISC1 cleavage, she saw that when she provided a homologous donor, some of the time, one side of the break looked like it was repaired cleanly, homologously as expected, and the other side was repaired by end joining. And this is with a linear double-stranded donor. And so maybe that's not surprising. You got resection on one end and initiation of homologous repair, but the other end is a double-strand break, you know, and it could be end joined to the other side of the induced break. So it's not so surprising. Randy gave the usual explanation for how they think that happens, and of course I had to produce the opposite explanation. So I think maybe what's going on is that the non-homologous joint is made first, particularly in fish, but also in mammalian cells. Non-homologous end joining is cranked way up. So the non-homologous joint is made first. And we know that, so even though the donor is single-stranded, we know there are some ligases that will join single strand ends to each other, or maybe a single strand to a double strand. And if the cut turns out to not be flush or is processed a little bit, maybe there's a way to efficiently make an end join. And then at a more leisurely pace, something's happening at the other side where some resection occurs and you can get annealing and then resolution.
But if that happened, then the donor piece is now part of the broken DNA. What is it resectioning to? What is the other end resectioning to?
So I guess what I'm thinking is that you make a break, and the single strand oligo gets joined to one of the strands at the break. And now on the other side, I better draw this in a sensible way. If I have a 50-50 chance here, right? So you can correct me if I'm wrong. At the other side, there's some 5' to 3' resection. And this donor, although it had homology on this side, didn't use it, but it still has homology here to that. And so that's the homologous joint on one side that they expected, and a non-homologous joint on the other. So it could be going that way. And it is true that in this case, the donor is at least partially physically incorporated, whereas the way we usually draw things, the 3' end on one side lifts up and pairs and then copies the donor, and then if this unpairs again, or if this now gets degraded, this goes back and hybridizes to the other end at the break, which has also been resected a bit. So in that case, there could be no physical incorporation of the donor. It's just a copying from the donor that was done. But in this case, there's some physical incorporation. And there is a group at the University of Minnesota, led by Eric Hendrickson, that's been trying to distinguish in cases like this with an oligo donor, whether the oligo is physically incorporated or just copied. I'm not sure it matters a whole lot ultimately, but I think that's the source of the one end homologous, one end non-homologous, is that the two processes are both using the donor, one on one end and the other at the other end of the break. What else? Are there any issues from during the week that you'd like to talk about? I may not know anything about them, but we could at least talk about them. Nope. So on behalf of all of your colleagues who came to the workshop, I want to thank you for being very engaged participants. I think it's one of the reasons it's worked out well is because you have been so eager and interested, and participants rather than just sponges sort of taking things in. You've been very active in your engagement with the workshop. So thanks for being here. Thanks for your participation.